All right, for our muscle slides. Now the thing to recognize here with these muscles, really we're just all about what causes the contraction to happen. All right. Now a key thing I want you to maybe kind of peek at here or look at is down into this outer layer, this sarcolemma of all those bundles. So I really liked this picture packet because it kind of helped me kind of get that good sense of scale of what I'm really looking at here in the diagrams of how those actin and myosin fibers are twitching. Um, so yeah, this kind of does a good job, takes you from your arm all the way down to these interesting things about a muscle fiber, a muscle twitch. And the thing I tried to emphasize, like I dumped a bunch of calcium in a jar or something in a jug. And really what you could see there is just, calcium's reactive, calcium's insoluble, but you have calcium stored in a lot of membranes and all your cells, I think. It's wrapped up, it's not really necessarily vesicles, but it's wrapped up in within the endoplasmic reticulum, and it's there. And then obviously you store a lot of calcium salts in your bones. There's calcium in that nerve. That nervous impulse, it actually releases some stuff like some acetylcholine is part of that. And I mean, there's a lot of things that can kind of affect it. But when the nerve gets there, what happens in the synapse is it triggers the calcium release that's stored in the ER of the muscle tissue. So once that calcium release happens, the muscle fiber, the muscle contraction is automatic. That tension that it creates is kind of a spontaneous reaction there. I know, I just thought, obviously all your muscles kind of function a little bit differently. I mean, eye muscles, they're going to have this tension that's much more exact, if you will, much more refined. The gastrocnemius, I think this is like the top part of your calf muscle, a little bit bigger range of tension there. But it's interesting that it's a similar strength. This is for each fiber that's in there. And then this is like the soleus, it's like the bottom part of your calf, kind of a bigger muscle, a bigger range, a bigger time to get all those fibers to contract. So obviously in this little way we look at it, I mean, each muscle is going to be behaving differently. And quite honestly, this is pretty much, I guess the way we look at it, this is more for striated muscle tissue. So cardiac muscle would look like this. Skeletal muscle would look like this under a microscope. Smooth muscle tissue would look different. And the other thing with smooth muscle tissue, I mean, it's under hormonal control a lot more than skeletal or cardiac muscles are as well. Just interesting things about muscle, as a developing embryo, you get these cells that, I guess pluripotent stem cells that could develop into muscle tissue. They actually fuse together. That's what a muscle fiber really is all these fused cells. Yeah, even in the ones we've had, I don't know how much time you spent, but I could see several nuclei. I could see this image here pretty clear under this tissue in our microscopes. So lots of nuclei. There's that outer layer. Now, if you peel out kind of one of those little fibers there, that's what we start to really kind of look at. Really, our knowledge of a lot of this, it's not necessarily studying people. A lot of animals have some, I think earthworms, we kind of looked at some of their muscles to kind of learn a lot about this. Okay, yeah, so that's just kind of the notes from class. Yeah, but we're really going to kind of look at this actin-myosin interaction and what happens there and what's floating around in the fluid in the sarcoplasm to affect this binding and contraction between actin and myosin. Okay, you want your muscles to contract, relax, contract, relax. You need them to be in the relaxed phase, ready to fire. If your muscles are always in the contracted phase, that's, first of all, it's not good. But that is the spontaneous, or when those muscle fibers, actin and myosin, touch, they naturally contract. All right, and so your body, kind of like the nerves, the role of our energy molecules, our ATP, is they're denaturing, they're phosphorylating that myosin hinge in order to maintain the resting phase of the muscle. So it's just kind of like you want your nerves ready to fire, you want those gates, and you want the sodium-potassium ion balance to be proper, to be correct, so it's ready to fire, ready to go. Just like your muscles, you want them in the relaxed phase, you want them ready to fire, ready to contract, ready to go, and ATP does that for both muscles and nerves. That's kind of why you say it keeps us alive. Okay. kind of like, yeah, so another picture is kind of same thing. The fluid is the sarcoplasm, the little fiber there that contains these thin and thick filaments is called a myofibril. All right, the sarcoplasmic reticulum, that's where a lot of the calcium salts are stored in there. The nerve hits the sarcolemma and causes, when that action potential gets there, releases, I don't know, there's a lot of different intermediates you could talk about, but ultimately it gets to this release of calcium that was stored in these sarcoplasmic reticulated membranes. I kind of like this one. It's like, okay, this is like the big arm muscle. Big muscle there. You know, you can like thinking, okay, I'm eating lunch. Oh, what? I mean, sometimes, I don't know if you get a piece of steak, like, oh, I pulled a vein, or oh, I pulled a little muscle fiber out. So yeah, you can kind of imagine, okay, you pull that out, then you start chopping that little thing up, start looking at it under a microscope. This here is the draw. I just see this, you see this, it's just kind of a way to represent on paper, is really what it is. But you see this kind of image a lot on these, just any kind of protein interaction question a lot, that protein interaction that results in movement, results in locomotion. So you can kind of imagine picking that little piece of steak out of your teeth, pulling it out, chopping it up, and then here, this picture here kind of looks like this, what it would drawing would be on paper. So yeah, let's kind of break down this, this motion of the muscle fibers. So here's that kind of way to represent it on paper. These purple ones with the little bumps, those are the myosin. Those are the thick filaments. The little bumps are the myosin heads. Here's a good picture of a myosin protein. The whole thing, the whole reason this works is that hinge. That's the part that gets denatured. That's the part that changes shape. That's the part that causes movement, especially when it's anchored or locked onto an actin fiber, which here are these. So if you think of this drawing, the dark regions under a microscope would be here, where there's a lot of overlap. Light wouldn't get through. Here would be the lighter colored, where the light does get through on a microscope. Now, I know, I remember in college we'd do these things. It's kind of like they gave us this, I hated it, but they gave us like this live frog. Like, okay, cut some muscle tissue out. Like, and you cut out those, and then you could look at it under a microscope and squirt like, different neurotransmitters. Nicotine would do it, calcium would do it and stuff, but you just start squirting stuff on the muscle fibers and they just kind of start spontaneously contracting under the microscope there for you. And you can see this overlapping region here get dark. Okay, so what's happening? What kind of yeah, here's a good picture. The contraction, you can see where here's like kind of relaxed, and then here that light region gets just gets smaller as those things overlap and as they contract. Part of the fascinating thing about me too is just how it's so well organized. Because look at that, you could pull out all these little fibers, pull them out, pull them out, pull them out, and the dark light is there. So it's very, very consistent, very organized where the overlap occurs throughout that tissue. Okay, so what happens here? How do things move? Well, we've kind of indicated that calcium really is kind of what allows it to happen. Let's look at what calcium's job is. So here's that picture. This is just showing those little bumps where they're kind of touching. Let's focus right here. This is a good place to kind of look at. Now, once they touch, the contraction is spontaneous. So to keep them from touching, they're kind of naturally attracted to each other. This tropomyosin and this troponin, these guys here that kind of wrap around the actin, myosin is not as attracted to that as it is to the actin, to that. So if these guys can get denatured, if these guys can change shape, if these guys here can move, as you see here, it exposes the active side of the actin. What allows these guys to kind of be denatured and to change shape? It's the calcium ions, calcium salts. Where did those calcium salts come from? From the sarcoplasmic reticulum, kind of all wound up in the membranes. But once that is exposed, these guys can touch. Once those guys touch, once they interact, once they touch, this is spontaneous. This here, this hinge will spontaneously change shape. And it's just kind of a side group interaction thing, a hydrogen bonding thing is a big part of it. There's some other kind of intermolecular forces that change as a result of that. But the net result is the hinge changes shape and the muscles contract. So you can kind of maybe visualize how everything would slide over. And the thing that makes it uniform and that makes it coordinated and that allows you to move your entire arm really is kind of that this calcium, it floods. It's like, oh, everywhere in the arm, everywhere in the thing you want to move, everywhere in that. So once that calcium starts moving everywhere, everything starts touching, everything starts twitching, and you kind of just experience, oh, I'm moving. Okay. So what are the steps here? Calcium comes in, tropomyosin and the troponin denature, they move out of the way. Actin and myosin bind. That binding spontaneously causes the hinge to change shape. That hinge changing shape is a contraction. Now, where the work comes in and what's really kind of needed here is that's what ATP is doing. ATP, and it's a constant source. It's not like, oh, I just got one little ATP. No, you need it phosphorylate, phosphorylate, phosphorylate. This hinge needs to be phosphorylated regularly in order to stay this way, in order to stay in that relaxed phase. And then the next time you think about something, the next time something triggers a calcium release, a contraction can happen. I know, just some interesting things like why do you cramp up? Why do you get tired? Why do your muscles not perform? I always wonder, it's like, oh, I'm thinking, I really want to move my muscle, I just can't. I'm really trying to, I just can't. So yeah, I mean, obviously ATP is a big part of that. So not enough oxygen, not enough glucose, cramps. You could have damage to that. I mean, so if this signal's not getting through to release the calcium, then you can't. And this, some genetic diseases cause this. Injuries. It's like, I tore the heck out of my muscle. I'm thinking about, I just can't move it. And that's because you lose all of that control. pH is a huge part of that. If the pH of that fluid changes, the way it's going to denature changes. So obviously that whole equilibrium and that very sensitive environment there is going to affect that. So the calcium really functions best at a very certain pH. If you're running out of energy and you're going into oxygen debt and lactic acid builds up in the muscle tissue, that affects the calcium binding, that affects your strength. That affects how many of those fibers will actually be able to fire and go. And really, it's just kind of a cumulative effect of all of those. I tore it up. I'm not eating. I'm not breathing. It's building up lactic acid. All of that together is going to affect calcium binding and affect that contraction. And just even getting hot. Working out used to get more blood flowing, smooth muscle around the blood vessels dilates. You start to get a little hotter. I really think of temperature as a pH change as well. That's kind of the net result of that. So obviously compared when you're ready to go, ready to compete, ready to lift, ready to run, that's when you've got good oxygen, normal pH, you're well rested, you've got a lot of energy supplies all kind of built up. Anyway, hey, thanks so much for the muscle function. I hope that helped you. I think next we'll do digestion. How does this nutrients, how do these metabolic reserves find their way into our tissue?